Bismillahirrahmanirrahim. BDS students, now this is lecture 3 and on the transcriptional process. The transcription, first we in lecture 1 and 2 we discussed about the transcription in prokaryotes, and now in lecture 3 we discuss the transcription in eukaryotes. In the previous lecture, we discussed about the process of transcription. Transcription defined as this is the process for the synthesis of RNA. It utilizes the substrate that are the nucleoside triphosphate. Ribonucleoside triphosphate utilized as the substrates for the transcriptional process. RNA polymerase, these are the enzymes which are the multi-subunit enzymes, and they are used for the transcriptional process both in prokaryotes as well as in eukaryotes. However, these RNA polymerases, they are of different types both in prokaryotes as well as in eukaryotes. Prokaryotic RNA polymerase, it consists of core enzymes as well as the holoenzyme. The core enzyme constitute 2 alpha, 1 beta, 1 beta prime and 1 omega subunits. Alpha subunit responsible for subunits SMD. Beta prime, it is responsible for template binding, and beta subunit, it is responsible for the 5 prime to 3 prime polymerase activity. The synthesis of RNA is always in 5 prime to 3 prime direction. The polymerase enzyme, it always reads the DNA template in 3 prime to 5 prime direction, but it can synthesize the RNA in 5 prime to 3 prime direction. Function of omega subunit, it is unknown, and other additional subunit which is known as the sigma factor. It recognizes, this is used for the recognition of a promoter region. These promoter regions, they are present at the beginning of the gene, and in case of prokaryotes, promoter regions box and -35 sequence, they are present. First of all, the sigma factors, they recognize the -35 sequence and form a closed complex. Then it also recognizes the box, which is centered at about 10 nucleotides from the upstream from the transcription start site, and form a open complex. Open complex means there is an unwinding of the two strands. Now, by the utilization of nucleotide triphosphates and the ATP, it initiates the chain synthesis. When it reach at about 10 nucleotides synthesized in the RNA, then it will clear the promoter and enter into the chain elongation. These chain elongation, when take place, however, when it reached the termination region, then it enter into the termination phase, which may be a row dependent. Row is a protein which is used to separate the RNA from DNA template, or maybe row independent. Here the stem and loop structures or hairpin structure is formed, as I have already discussed them, and by these structures, they separate the DNA strand. This is the transcriptional cycle in case of prokaryotes. The promoter regions, which are the consensus sequence like box as well as the -35 sequence RNA polymerases, they recognize them through the sigma factors. However, when these promoter regions, they change in different genes, like different genes, each gene has their own promoter region, and when these genes they change, then they require the different sigma factors. This is known as the sigma switching. Now comes the transcription take place in eukaryotic genes. Eukaryotic gene, it is much more complicated as compared to the prokaryotes. So the transcriptional process, it is also very complicated in eukaryotes as compared to in prokaryotes. In prokaryotes, only one polymerase it can synthesize the whole types of RNA. But in case of eukaryotes, separate polymerases they are utilized for the synthesis of ribosomal RNA, tRNA, and messenger RNA. These they are the different types of RNA, and even in small RNAs. So different polymerases they are used for the synthesis of all these different types of RNAs. Here the transcriptional factors they are also involved, and the transcriptional factors they bind to a specific consensus sequence or means specific promoter regions, which may be a close or proximal or maybe some distance from the transcription start site. And the assembly of a transcriptional complex at the promoter region determine which type of gene to be transcribed. So each gene has their own promoter region, own regulatory sequence, specific regulatory sequence, and they also bind the specific transcriptional factors whenever to be transcribed. One of the complicated phenomena for the transcription or for the transcriptional factor to access to the DNA is the remodeling of chromatin, and this phenomena allow the transcriptional factor to access the DNA. If we look at the chromatin structure and then the gene expression, then we see that in chromatin there are nucleosomes present in eukaryotes, and these nucleosomes they also affect the transcriptional machinery so as to access to DNA and transcribe a specific gene. Euchromatin form of chromatin is that which is the relaxed form, mean they contain less nucleosomes, they are not condensed so as they give the ease for the transcription factor to access the promoter region. However, heterochromatin they are highly condensed form of chromatin and contain a large number of nucleosomes. So mostly the inactive segment of DNA which is less transcribed or not transcribed, they are present in the form of heterochromatin. However, these can be changed, these forms can be changed or interconvertible to each other. Heterochromatin may be changed into euchromatin by the process or by the phenomena which is known as the chromatin remodeling. Chromatin remodeling is the phenomena by which the heterochromatin it can be converted into the euchromatin, or euchromatin by heterochromatin by a specific or through a specific enzymes. Acetylation is the phenomena, it is the post-translational modification of the proteins, the histone proteins which contain the lysine residues at the amino terminus. They undergoes the acetylation through which the interaction of these histones to the DNA there are weak interactions so as to you can say that conformational changes and access of transcriptional factors to the by decreasing these interaction. This is one of the phenomena in regulation of gene expression. Now the transcription factors they can easily access to their specific promoter region. Another enzyme which is known as the histone deacetylases. Histone deacetylases they restores the positive charge, and again there are stronger interactions. So the chromatin remodeling take place when the euchromatin has been changed into the heterochromatin, or heterochromatin form may be changed into the euchromatin by means of this acetylation phenomena. This is the acetylation. How acetylation? These are the lysine residues, the R chain of the lysine residue. Here, by means of histone acetyl transferases, they acetylase this amino residue present on the lysine residues. But histone deacetylase enzymes, they again restores the positive charge. Here, loss of the positive charge, and there is again restore of the positive charge. Nuclear RNA polymerases, there are the three different classes of nuclear RNA polymerases in eukaryotes. These three classes, they can synthesize all types of RNAs in eukaryotes. Basically, these enzymes, they are large enzymes, and also they are the multi-subunit enzymes. Each type of RNA polymerase, they can recognize a particular type of genes. It means that these particular type of genes contain or carry their specific set of promoter regions. So each type of RNA polymerases, they are specified for their specific promoter regions which are present on a specific genes. These three types of RNA polymerases, they are RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. One of class is RNA in eukaryotes is RNA polymerase 1. RNA polymerase 1, it synthesizes the precursors of R28S, 18S, and 5.8S ribosomal RNAs. Basically, the ribosomal RNAs, they are synthesized in their precursors. Nascent forms are the precursor which then modifies to form a functional RNAs. After modifications, which is known as the post-transcription modification, we later discussed these post-transcription modification. These precursors, they are modified into a functional transcript. Next class, this is the RNA polymerase 2. RNA polymerase 2, it is the it synthesizes the nuclear precursor of the messenger RNA. RNA polymerase 2 synthesize the coding RNA, which is messenger RNA. Coding RNA mean it is further translated to form a protein. So it can also synthesize the small ncRNAs and nuclear RNA, snRNA, nucleolar RNA, as well as the microRNA. The main function is to synthesize the precursor of messenger RNA. Messenger RNA is also synthesized in its precursor form. The nascent form is long as compared to the functional form. After then, it undergoes the post-transcription modification by which this precursor is converted into its functional form. Now, the RNA polymerase 2, which is responsible for the synthesis of messenger RNA, which is a coding RNA and responsible further translated to form a specific type of protein. This RNA polymerase 2 has specific promoter region in eukaryotes. One of the promoter region in certain gene, in some gene, is the Hodgkin's box or Tata box. Tata box or Hodgkin's box present in some genes, and Tata binding protein, they bind. These are the basic transcription factor which bind this box so as to initiate the other transcription factors come bind to their specific regions, and also the our transcriptional machinery so as to form a transcriptional complex. One of the promoter region in eukaryote is Tata or Hodgkin's box. It lie about 25 nucleotides upstream of the transcriptional start site. 25 nucleotides from transcriptional start site. Next is the another promoter region is the CAAT box, CAAT box. It lie at about between 70 to 80 nucleotides upstream from the transcriptional start site, and mostly the constitutive genes. They are expressed through the Tata box. They are expressed, which don't contain the Tata box. They typically present the card box. The constitutive gene, which doesn't, these constitutive genes, they don't contain the Tata box mostly, and they typically bind with this card box. Another region which is known as the GC rich region, they may be found in certain genes. So there are the different types of promoter regions, and which type of gene has to be transcribed, their specific transcription factors, they come and bind to their specific regions, to their specific promoters, so as to transcribe the specific gene. This one is the eukaryotic gene promoters consensus sequences, which are present at the transcriptional start site. Here you see the transcriptional start site. This one is the Hodgkin's or Tata box present here, centered about at this. This one is the cart box. Then further upstream to the GC box. So about 25 bases upstream from the start of the transcription present the Hodgkin's box or Tata box, and this Tata box contains some of the gene, but about 70 to 80 nucleotides upstream from the transcription start site. This one is the transcriptional start site. 70 to 80 bases upstream from this transcriptional start site lie a cart box. Mostly the constitutive genes, they contain this cart box. These are the different promoter regions present in the eukaryotic genes.